This is the module for case study for methods in neurobiology. This case study is titled Genetically Encoded Serotonin Indicators for Assessing Panic-like Response in the Prefrontal Cortex. The background of this study concerns panic disorder, which is a psychiatric condition that's characterized by recurrent and spontaneous panic attacks that can impair daily functioning. There are two main theories for the cause of panic disorder. Um, the neuroanatomical theory of panic disorder was developed in the 1990s by Jack Gorman, and it um, essentially focuses on the brain structures that are involved in the regulation of anxiety and fear. So there's a complex network that includes the medial prefrontal cortex and various subcortical structures that regulates anxiety and fear. This subcortical structures such as the amygdala, the hippocampus, the hypothalamus, thalamus, the anterior cingulate cortex, the locus coeruleus, and the periaqueductal gray region are are part of that network. The image on the right shows some of the larger structures in that network. At the top, we have the cartoon of the human brain, and then below is a cartoon of the rodent brain, and it just is showing the homologous structures. So in this presentation, we'll mainly be concerned with the medial prefrontal cortex. As you can see, um, the that the prefrontal cortex in the human has a homolog in the rodent brain. Um, so the neuroanatomical theory basically states that panic disorders are a result of a dysfunction in these circuits. The second major theory is the neurochemical theory of panic disorder, and that was developed by J.F. Deacon and Federico Greff, um, two medical psychiatrists, researchers, and they developed this in the 1990s as well. First paper was in 1991. And basically, it states that serotonin plays a major role in regulating anxiety and fear, and that panic disorder is a result of dysfunction in the serotonergic systems. Prior to their paper, it was known that monoamines that include serotonin, norepinephrine, and dopamine contributed to anxiety and depression. But this theory focuses on the serotonergic aspect. We know serotonin plays a major role because selective serotonin reuptake inhibitors are the first line treatments of panic disorder, but it's not quite clear how they work. It's not completely understood. Um, we know that SSRIs reduce the endocytosis of serotonin back into um, the main neurons, mainly from the dorsal raphe nucleus, but we're not quite sure how it works. The problem statement for this study um, is that the medial prefrontal cortex regulates fear and anxiety via inhibition of the amygdala and through other subcortical structures. Um, but the underactivity of the medial prefrontal cortex is linked to panic disorder. The the raphe nuclei constitute the major serotonin pathways in both the human brain and in the rodent brain. And you can see the image on the right. You can see the various raphe nuclei in the aqua blue color, and then they have projections that run out through the brain. That shaded purple region represents the ventromedial prefrontal cortex in humans. And at the bottom, you can see the medial prefrontal cortex in a rodent, which is the homolog that we discussed earlier. The dorsal raphe nucleus is the main nucleus of the serotonergic system, and it densely innervates the medial prefrontal cortex. So it's known that serotonin produced by the dorsal raphe nucleus modulates the medial prefrontal cortex in some kind of manner, but all the mechanisms aren't completely understood. Um, in the initial research on serotonin, it was somewhat confusing. So there were some models that suggested that serotonin excess results in panic. There were some models that suggested that a deficit of serotonin caused panic. Um, and there are other theories that propose that serotonin kind of has a dual role, that it can inhibit panic, but increases generalized anxiety. So again, that's that contrasting fear state versus anxiety state. Um, the modulatory effect of the dorsal raphe nucleus on the medial prefrontal cortex and anxiety and fear regulation is still not completely known. It's one of the areas that's still being explored. So therefore, real-time measurements of the serotonin dynamics in the medial prefrontal cortex during panic are needed to further understand fear regulation, and through that, understand panic disorder and the medicines that are used to treat it. The solution and the tools. Um, exciting new work was done by at the University of Bremen, and that involved a new um, marker for serotonin, and this was done by um, Martin Kubicki at the University of Bremen and his colleagues at the German Center for Neurodegenerative Diseases. So this new molecule is called sDarken, and it's part of a new family of genetically encoded serotonin sensors that allow real-time in vivo imaging of serotonin dynamics. And sDarken is a genetically encoded indicator. It's a genetically encoded transmitter indicator. And the key features of sDarken are that it modifies the 5-HT1A receptor, which is expressed in the prefrontal cortex, and they modify that receptor with a GFP reporter. The sDarken sensor is bright in the unbound state, but when it's reduced, it's fluorescence with reduced fluorescence upon binding of the serotonin. So you can see in the upper right, when the serotonin is unbound, the reporter is illuminating, and when the serotonin binds to the 5-HT1A receptor, the fluorescence decreases. It's a highly sensitive and selective reporter, and it has high temporal and spatial resolution. And it has already been demonstrated in the detection of endogenous serotonin release in the prefrontal cortex of mice. So that was a key feature that's already been proven to be useful for imaging. Um, the image in the lower right shows repeated application of 5 millimoles of serotonin in 9 cells from the paper by Kubicki and his colleagues. And what that really shows is that over a 600-second interval, when the serotonin is applied, there's a drastic decrease in the fluorescence signal. So it really lowers when it binds the receptor, and then gradually increases after the serotonin is they quit applying it. The other tool is a 2-photon microscopy that enables real-time observation of the dorsal raphe nucleus derived serotonin release in the prefrontal cortex during panic-like events. So the research aim and the hypothesis. So the aim is to determine real-time serotonin dynamics in the medial prefrontal cortex during odor-induced panic-like states using 2-photon imaging and the sDarken sensor. The sDarken sensor is just has a high affinity for serotonin, and it's brighter. The hypothesis states that panic onset will cause fluctuations in serotonin release in the medial prefrontal cortex. Panic states will be associated with serotonin, while non-panic conditions will show increased serotonin. So the research plan first consists of obtaining 10 C57BL/6 mice. These are standard black mice used in research, roughly 10 weeks old. Studies have shown that the murine medial prefrontal cortex is homologous to that found in higher order mammals, as we showed earlier in the first two slides. The second step involves AAV sDarken and being injected into the medial prefrontal cortex for sensor expression. As mentioned, the sDarken sensor is a high affinity sensor variant, and roughly wait 4 weeks for the gene expression. And step 3 is to create a cranial window above the medial prefrontal cortex in each mouse and allow 3 weeks for them to recover. And the first key step was to obtain a baseline imaging. And this step involves placing the mice on a running wheel and fixing the head under a 2-photon microscope. And placing the mice on the running wheel relaxes them and gives them something to do as they're secured. Their head will be fixed in the microscope, as shown there on the image in the lower right. The serotonin signals will be recorded for 30 seconds while exposing the mouse to neutral odors. Repeat for each mouse to establish the baseline. And the excitation wavelength for the sDarken indicator is 920 nanometers, while the GFP emission wavelength is between 500 and 550 nanometers. And the next stage is to induce panic. So the panic can be induced in the mice by exposing them to TMT, which is a synthetic predator odor. And this sequence involves 20-second exposure to the TMT to elicit a fear response. The TMT is removed, and then the neutral odor is presented for 120-second recovery period. So the serotonin dynamics are recorded for the entire 120-second period with 2-photon microscope, and this repeated for each mouse. So just to clarify, that 140-second interval includes both the panic phase and then 120-second recovery period. Number 6 is to obtain the test data. So that involves two main parts. One is to observe the behavioral markers of the mice that relate to fear. So during the panic induction phase, freezing, which is, mobilization behavior, the running speed and pupil dilation will be observed and recorded. The delta F over the F signal will also be calculated for the 120, it should be 140-second panic recovery trial. This should be repeated for each subject. And the second to last step is to obtain the control data. So using the sDarken expressing mice, they record a 2-photon signal from the medial prefrontal cortex while exposing rodents to a neutral odor. This will be used as the control. The final step is to analyze the data, compare the serotonergic dynamics in the medial prefrontal cortex between panic and neutral conditions, and compare and interpret changes in signal amplitude, timing, and recovery relative to the mouse behavior. And the expected results and conclusion. So the expected results from the hypothesis are that with panic induction, the serotonin in the medium prefrontal cortex will decrease significantly, exhibited by a steep drop in the delta F over F signal. The neutral conditions, serotonin levels remain elevated or stable. In post-panic recovery periods, serotonin levels will gradually increase and approach the stable baseline conditions. And the serotonin dynamics are expected to correlate with the freezing behavior, running speed, and pupil dilation. Those are the fear behavior markers in the mice. So conclusion, real-time 2-photon imaging using sDarken serotonin sensors elucidate the role of serotonin in the prefrontal cortex during panic onset. Such techniques can clarify the therapeutic mechanism of SSRIs in the treatment of panic and anxiety disorders, and real-time imaging of other brain structures using serotonin sensors could lead to new and improved pharmacotherapies for panic disorder and other related anxiety disorders. There's other references. Thank you for your time.